Thank you for joining us for today's webinar, Doing More with Less, From Genotyping Arrays to Low-Pass Whole Genome Sequencing, presented by Dr. Elizabeth Louie and Dr. Eric Yeager. Our first speaker is Dr. Louie, supervisor of the Technical Applications team at GENEWIZ, who received her PhD in Molecular and Cellular Pharmacology from Stony Brook University. She provides consultation to customers across all of GENEWIZ's genomics solutions, including next-generation sequencing, DNA synthesis, and any custom molecular biology needs. Our second speaker is Dr. Yeager, staff scientist of the Emerging Applications team at Illumina, who received his PhD in genetics from the University of Chicago. He is a 14-year veteran of Illumina and has led assay teams responsible for the development of a wide range of Illumina products, from the Golden Gate bead chips to the original TrueSight tumor assay. Without further ado, I'll hand everything over to Dr. Elizabeth Louie and Dr. Eric Yeager to begin their presentations.
I think it really just depends on someone's project goals and exactly what they're looking for. As a quick overview of the topics that will be presented, I'll first go into some well-established genotyping technologies. Then I'll give an overview of what low-pass whole genome sequencing is. We'll review some applications of low-pass sequencing, and then I'll present the main takeaways of the session. So let's start with some introduction to genotyping technologies. In order to understand how far new technological efforts in sequencing has come, I'd like to call out back to nearly 20 years ago. This was when the initial results of the Human Genome Project was first published. It was a culmination of 13 years of work, around $2.7 billion spent, and 20 groups worth of effort. It covered 95% of the genome, and the process was performed using traditional Sanger sequencing, involving the generation of back clones, identification of overlapping clones, fragmentation, then sequencing, and assembly. This breakthrough launched a new field of genomics that many of us now take for granted, from transcriptome profiling to the generation of novel DNA clones to gene editing. And one of those notable advancements was the creation of technologies that used hybridization, like DNA microarrays. Now, DNA and genotyping arrays are a cost-effective method to detect genetic variation in highly similar sample sets. It's a great alternative to what at that time was a costly and laborious effort to sequence entire genomes. And while limited to variants derived from existing data, there were many benefits that outweighed that limitation, such as the overall cost, the speed, and the accuracy. Microarrays allows for studies of thousands of patients' worth of data, has a high throughput, and can efficiently detect known SNPs at a very high accuracy. However, there remain some caveats. Since the method is heavily reliant on that existing data, arrays have been biased to European populations. Despite this, genome-wide associated studies have continued to grow, though the focus is still heavily targeted to one population. Therefore, while there are time, cost, and accuracy benefits to using arrays, the applications of these studies are limited to better studied populations or to the detection of those known variants. And these limitations can only be overcome through DNA sequencing. Now, the cost of sequencing has continued to decline since the Human Genome Project completed, with significant strides with the introduction of high-throughput screening options like next-generation sequencing. Notably, costs dropped significantly with the introduction of the first Illumina platform. And while the cost has gone down due to technological advancements, the number of sequencing publications accordingly increased, with 3 exemplar projects in particular. One, the 1,000 Genomes Project, which documented hundreds of human genomes. 2, the Cancer Genome Atlas, or the TCGA, which documented several thousands of tumor normal genome pairs. And the last, the Exome Aggregation Consortium, which documented over 60,000 human exomes. Now, many of the data sequenced to date has been whole exome rather than whole genome. As advancements are continually made, though, we expect the ratio to be increasingly favored towards whole genome in the future. Now, all that said, while traditional whole genome sequencing overcomes the limitations of arrays, and while the decrease in cost and speed has driven adoption, it does remain price prohibitive for population studies for most researchers and most groups. And because of this, it's important to understand the pros and cons of certain DNA sequencing workflows so that researchers can weigh which option might be the most beneficial to them based on their project goals. Now, to start, I'd like to review the advantages and disadvantages of a whole genome sequencing approach, as well as using targeted sequencing, like whole exome sequencing. So, whole genome sequencing remains a great option that answers a variety of research questions. It allows detection and analysis of samples without a known or complete reference sequence. It also provides detection of different abnormalities beyond a simple variant detection, and that includes structural rearrangements, copy number analysis, and other types of analyses. Appropriate applications would be anything that requires that discovery element, or where a reference sequence might not be unavailable, such as biomarker discovery, contamination testing of products, or any novel organism studies. On the other side, there are several different targeted sequencing methods that can either supplement or be an alternative to whole genome. This can be targeted through a traditional PCR amplification assay, or it can be a probe or bait hybridization method that enriches for a region or regions of interest. One method would be chosen over the other based on a few different characteristics. One, the length of the region or the regions of interest. Two, the availability of unique sites for primer binding. And three, which is typically a more general consideration, the scope of the project and whether one of the methods being introduced or being used would have an unintended enrichment bias due to that technology. And in either case, unlike whole genome sequencing, the design would require a known genomic sequence. Targeted sequencing has many benefits. It provides the ability to sequence low-frequency variants with deep coverage with a lower output compared to performing the traditional whole genome approach. It's robust, and it also has focused and easily interpretable variations for disease models to inheritable disease. Now, the data is often a more focused data set. So, for example, 2 to 3% of the human genome encodes for a protein, and many studied disease biomarkers or models are linked to mutations within that coding region. So with whole exome sequencing, or any type of targeted approach, researchers can hone in on the relevant regions for sample comparisons instead of going through the entire genomic data. But if you also look at it from another way, only 2 to 3% of the human genome encodes for a protein. So the remaining 97 to 98% would not be captured in a traditional exome or even a hotspot cancer panel assay, which is really heavily geared towards coding regions and regions of specific genes. So a targeted method cannot detect abnormalities across the entire genome like large-scale substitutions, deletions, insertions, any epigenetic changes, and any chromosome inversions or translocations. Therefore, to summarize the limitations, exome sequencing is still limited to the current understanding of the genome and exome and is limited to the types of mutations it's able to detect, while whole genome sequencing can address all of those limitations of that targeted approach, but would require that same amount of coverage and therefore increasing the amount of data output required. The deeper sequencing requirement does limit the feasibility of these high-resolution large cohort studies. So that brings us to the main topic of our discussion, low-pass whole genome sequencing. So low-pass whole genome sequencing is a high-throughput solution that's comparable to a traditional genotyping method like an array. With sequencing at very low coverage, anywhere between 0.4x to 4x for most studies, it also uses imputation to generate those variant calls. So during the Illumina portion of today's talk, there will be a more detailed look into how imputation works. But as a general overview for now, imputation uses statistics to make variant calls and can transform that data to provide up to 99% accurate calls across that genome. So let's take another look at how imputation from low-pass data compares to imputation from arrays. In a study released last year, a group of researchers performed testing of several conditions to look at two populations, individuals from a historically more researched European ancestry in an association studies, and then compared it to individuals of African ancestry, which is historically less common in association studies. For experiment A, 120 unique individuals were sequenced in triplicate to a target coverage of 0.5x. In experiment B, 120 individuals were sequenced in triplicate to target 1x coverage. Experiment C, one sample was sequenced 30 times to target 1x coverage. And for experiment D, 60 samples from the set of the original 120 were then sequenced to 1x coverage. The last experiment, experiment E, is for the 120 unique individuals being assayed in triplicate on a global microarray. Now, the results on the left figure show that the non-reference concordance across the different experimental groups and that the genotyping concordance is an important metric that quantifies imputation accuracy. So it shows that the low-pass sequencing plus the imputation provides accurate calls at effective coverages of that 0.5x and higher. And the results were comparable, or in the case of the African ancestry group, potentially even better than the array data. So here's a side-by-side comparison of the different methods we have discussed so far. So when it comes to novel SNP detection, low pass is a practical method compared to whole genome and whole exome sequencing. For the human genome, imputation allows for interrogation of 85 million SNPs for variant detection. And structural variant detection relies on good coverage across the entire genome, so low-pass coverage might not be ideal. However, copy number variant detection is still possible with the shallow read depths. Therefore, some of the highlights of low-pass is that it does allow for a high-throughput processing due to the low data requirements, and remains an unbiased method when it comes to detection, allowing for off-target analysis and any novel SNP detection. Therefore, low-pass sequencing is well suited for genome-wide associated studies and is especially advantageous over arrays when it comes to underrepresented populations. Low-pass can also be used for phylogenomics, any breeding studies, and that includes animal and plant, any cell bank profiling, and at the clinical level, any patient testing for a risk or outcome assessment. On the other side, low-pass should be avoided for use in genomes that are not well characterized or for any de novo assembly. And this is all due to requiring historical data for imputation. And while it can be used to detect SNPs, the low coverage is not ideal for low variant frequency detection studies or for data that will be used to make a call on a patient disease diagnosis. So now I'd like to highlight two publications that show how low-pass can be used to supplement and fit into anybody's study design. So this case is from the Broad Institute and was designed to test how effective low-pass sequencing and microarrays were for those underrepresented populations. As mentioned earlier, since most arrays are modeled off of results from European ancestry, there are many variants from other ancestries that will be missed. So the group here took data from another sequencing experiment where a deeper sequencing was performed on 91 individuals from Ethiopia, Kenya, South Africa, and Uganda, and downsampled the data to be comparable to low-pass coverages of 0.5x, 2x, and 4x. This data was compared to results of different arrays at low, medium, and high density arrays. So overall, the imputation accuracy is affected by the overall sequencing coverage, but has comparable concordance to several different arrays. Now, that said, the low-pass option did allow for capture for additional variants that were not available based on the array design, which provides an advantage to the study of this specific population. Next, I'd like to highlight a study coming out of Genentech, which was testing the effectiveness of using low-pass whole genome sequencing as a non-invasive method to monitor patients with lung cancer. So here, they explored the copy number variants of tumor-derived cell-free DNA to link to how well they performed in the clinic. In this study, 152 patients with non-small cell lung carcinoma were treated with several different therapies, and researchers isolated cell-free DNA from the plasma and performed low-pass sequencing anywhere between 0.1x to 1x. So low-pass was then subsequently followed up for a deeper sequencing, depending on how the variants results were. The results were then used to analyze the general tumor fraction with plasma based on the results of the low-pass whole genome sequencing results. And as a comparison, sequencing was also done using a matched tumor tissue whenever it was available. The higher sequencing depth did have better resolution and lower background for detecting genes and SNP variants, and the study itself concluded that around 0.5x was the ideal coverage that provided visible results while minimizing the overall sequencing depth. Based on the samples that had the highest tumor fraction, that's when the whole exome sequencing was performed to further explore the mechanisms that may have been involved in the resistance to that treatment and thereby causing a higher tumor fraction. So this study was really fascinating, and the points I've mentioned here are only a snapshot of the total work that they were doing. I did, however, want to emphasize that cell-free DNA is often very fragmented and has very low yield. This group and other groups have used low-pass successfully in predicting patient prognosis. So it's one of the many techniques that we hope will eventually be translated into more common use, even if it's only done as a predictive screen ahead of a more established technique in the clinical setting, like that targeted approach or like that whole exome approach. Which really brings me to what we at GENEWIZ call low-pass whole genome sequencing plus. Now, bundling low-pass whole genome sequencing plus a targeted approach at deeper coverage can be advantageous to just performing deep sequencing alone. Specifically, this works well with large studies where performing deep sequencing may be cost prohibitive on a high volume of samples, and the design requires a discovery element for new SNPs or similar, where a targeted approach alone or an array option may have that inherent bias. The second half of the talk will go into advantages of performing deep sequencing, but at a high level, it does help with validating the imputation data while allowing a more focused interpretation of a potentially smaller data set after that initial screen. So in summary, low-pass whole genome sequencing is one of the many techniques that are available for genotyping or profiling studies. And when thinking about which techniques to use, we do need to think about what the project goal is and what type of historical data is available to draw from. For low-pass, you need to think about if there's a database available for imputation, what types of variants or changes you're hoping to capture, and lastly, how large the study is. Low-pass whole genome sequencing is really specific to high-frequency variants and is really most appropriate for large-scale population studies that require this novel detection. And for designs that fall outside of that, one of the other methods discussed today might be a more appropriate option. And that concludes the first part of today's discussion. Please stick around because the second part will provide a more detailed look into low-pass whole genome sequencing and the technology behind it. But I wanted to thank everyone for their time and attention. I'll be around to answer any questions you might have after the second talk. And I'll go ahead and hand the reins over to Dr. Eric Yeager from Illumina. Thank you.
Okay, for this portion of the talk, then we'll be looking at low-pass whole genome sequencing and how the imputation derived from that can be improved through the addition of targeted deep sequencing. So just to highlight the 4 sections we'll be going through here. Again, we've already heard quite a bit about low-pass whole genome sequencing in the previous talk, but I will recap just a little bit of the highlights there. And then we'll take a look in more detail at how exactly imputation works, what benefit then it can gain from additional targeted deep sequencing, and then leave room for questions and discussion at the end. So again, what is low-pass whole genome sequencing? It's really just typically referring to sequencing the entire genome, but rather than at your normal 30x coverage, you're looking at much lower coverage from 0.5x to 4x, again, depending who you ask. It can vary according to what your particular needs are and the cost per sample that you're looking at, because obviously going from 0.5x to 4x is going to be an 8-fold increase in cost. So you do get more out of that as you sequence to higher depth, but there are diminishing returns, and we can take a look at that in some later slides. But the basic idea is then once that you have that low pass coverage, you can use a reference data set to infer or impute variants that may not have been picked up in that particular low pass data. And so in the end, you're essentially doing genotyping by sequencing. This will include both SNPs and also small insertions, deletions. So there are really two ways of doing this. The first is probably the most common, where you sequence the individual samples, as we're talking about with low pass, and then you go through this existing reference data set to impute the calls that you've missed. Alternatively, you can do a very large cohort of samples and aggregate the low pass data from each of them to create essentially a proxy reference data set that's used for imputation. It's a little bit more complicated. I won't focus on that particularly today, but it is an alternative if you don't have what you feel to be an adequate reference data set for your particular cohort. So what are the things to consider when you're putting together this low pass study? First of all, obviously, is how well are you able to call common variants? So there are a variety of tools out there for this, as we've seen previously, but it's important that you have confidence in those calls that are being made. And again, this in part goes back to the coverage depth that I was talking about previously. You can also think about how important it is to detect less common variants in your population. And by less common, I mean generally less than 5% allele frequency in the overall population. So is it really important to you to get down to those variants that maybe aren't seen as often and may be less prevalent in your reference data sets? And again, because then we're looking at a large amount of data, obviously there's going to be a computational informatic burden, which depending on your resources will come at some cost. There are companies that can do this for you, again, but that's something that needs to be taken into consideration when designing your study. Then at the end of the day, what is it that you want to achieve with the study? Is there a clinical or other application that it can be applied to? And we'll take a look at sort of the different amounts of data required to achieve different study goals, depending on your particular application. So again, just to emphasize that the real value of low pass whole genome sequencing comes when you do imputation. Without imputation, there's really not much that can be done with this data at such low coverage in your genome. And the power of imputation then really relies on the three questions listed on the right here. First, what is the size of the reference data set? Do you have many samples that have a broad representation of different genotypes present in the population that will allow you to get accurate imputations? So today, we're sort of on the left end of the spectrum where these large reference data sets number in the thousands, in some cases. And we're moving very quickly as sequencing obviously decreases in cost. We're moving to more and more of these reference genomes that can be added into the data sets. And this allows you then to detect more rare variants as you go. So in the future, when we've put together just millions of people at this point that have been sequenced, then you're in theory going to be at practically 30x quality of genome coverage, because you know all the different variants that are out there, and you can guess those few that you may have missed in your low pass data. But there are a couple other considerations too when doing this, because you want obviously your particular ethnic population to be represented well in that reference data set. Obviously, if you have one ethnicity that you're studying and the reference genome set does not contain many members of that, or a different ethnicity altogether, then you're going to not have the necessary alleles represented in the reference. So your ability to impute will be severely limited. So you can see that represented just generally by the the red, blue, and light blue boxes and the different populations to the left there on the screen. And finally, you want to consider what is the quality of the reference data set. Obviously, if these are older genomes, if they're maybe not at 30x coverage or other issues with the sequencing, if you have a lot of undetermined bases or ends that are listed in your genome sequence, then obviously you're going to be limited in what you can call using alleles from those particular examples. So the better quality data you have, obviously, the better quality your imputation. So with that, how does imputation work exactly? And it's not really magic, it's just a lot of math at the end of the day. So I've been throwing out some terms. Alleles, obviously, are what differentiate all of our different human populations. And the haplotypes then are these collections of alleles or different variants that are associated with other variants in that particular individual across their entire genome. So we see some different populations just represented, generally speaking, to the right here. And you can see African, European, Asian, each has its own sort of unique set of SNPs that are commonly involved or associated, I should say, with the different members of the population. So, for example, you see in your European population, for example, you've got G's, C's, G's at a given position across virtually all members of that group. But if you look at the African or Asian populations, then those particular variants are not present. Instead, you've got, for example, in the Asians, T's, G's, and A's at slightly different locations, and that is specific to the Asian population. So just again, emphasizing the need to have all of these represented, or at least the particular population of interest that you're going to be investigating represented in your reference database. And what makes now things start to get interesting is the linkage disequilibrium that's associated with these different loci and what has led essentially to the differentiation of these populations over time. So in some cases, you will have particular variants, for example, the A's and the C's in the first couple lines of the African population that are always going to be associated with each other. Occasionally, you will have some recombination event, as in the lines below, where you lose that C and instead end up with a different variant, now the G to the left of the A. So these events obviously occur over time and in great numbers, and this is what creates new haplotypes and, as I said, introduces new variants like the G with the A in particular subpopulations within your ethnic group. So based on this huge collection of variants that you have across all these different populations, you can see the diversity coming out as you get more and more of them. You can start to statistically infer which variants you're looking for in your low-pass data that aren't actually covered. So, for example, if we take the first line of the African population again, if you have captured that A, C, and G, and then you can essentially say that now we must have, based on what we understand of our population, the T, and the A that follow down the genome there in that particular haplotype. Because the other members of the population then are lacking those particular variants, and the C distinguishes them from the bottom half of the cluster there, where you have instead the G leading off. So these obviously are going to be a different group, and if you have that G and A and a G, then instead, okay, rather than that T, we're going to see a T, an A, before you get to the end of that haplotype. So this is really the nuts and bolts of it all. And again, as you can see, it's just power, diversity, and numbers, and then statistics at the end of the day. So as I said, when we start to get more and more genomes and more of these individuals represented, then the ability to do accurate imputation improves, and that's really where we are today now that the cost of the genome has come down enough to enable that. So I've been talking about variants that are common and variants then that are less common. So we talked about the ones in the colored boxes that are highlighted here. And these are ones that are present in most members of the population. But if that's all that you have, again, just to emphasize, you're not necessarily going to be able to differentiate those members of the population because everyone has them. So if you don't have that G or the C in the African group, then you can't tell the difference between the first 3 individuals and the next 2 individuals. So you can still do some things with that, but if you want to start to identify those less common variants, then you need to find another way to get at that data, because that's what's going to really give you the most complete view of a given haplotype. Again, not to undervalue these common alleles, because at the end of the day, they are present throughout the population in very high frequencies, maybe not 100% as represented here. But at the end of the day, you can still use those absolutely to calculate polygenic risk scores and other sorts of things, which we'll get into at the end of the talk. But that's one application, and we're looking potentially at more. And with imputation, really, again, you need those less common alleles to really start to differentiate individuals. And that's what this slide emphasizes. So again, just to drive home the point, once you have those less common alleles, then you can really start to separate out what's going on in your population. So again, we've got a couple of those boxed up and colored here. You can see the G's and the A's in the African population, for example, that now are very clearly showing what that particular haplotype contains and how it is different from the ones above it. So in the genome, you've got many variants that are present in less than 5% of the population. And depending on the population that you're looking at, can account for 85% or more of all variants observed in that population. So they are very valuable. Obviously, you're missing out on a lot of diversity if you don't capture them. So there's a lot there to be gained. But again, if they're not in your reference population, then your chances of detecting other ones associated with them are going to be limited. So you can also be missing out on quite a bit. If we take the exome as an example, if you look at those alleles that are found now in 0.1% of the population, so very rare, you're only looking at about 3% of the total number in the population. But overall, again, as I was saying with the genome, you've got 97% of all population variants. So again, just lots of information that's contained within the exome alone. And these numbers actually are probably a little dated. This is from the EXACT database, which has now been replaced with the Nomad database. So those numbers are even continuing to grow to this day. So capturing those rare alleles really does add a lot of power to your imputation at the end of the day. And this is, again, just to make it absolutely clear that these are rare alleles that are associated with rare haplotypes that are specific to a given population. So we've added in a few more here that you can see going down the list. It's true of every population. And again, the more sequencing that you get, the more individuals you have sequenced, I should say, the more robust your low-pass data will be when it comes to imputation. There obviously would be a variety of applications here. If you're looking at one particular ethnic population, as I said, then that's the population that you want to be seeing most in your reference data set. But if you're looking at a general disease across populations, then all those populations should be present in your reference cohort. So these are the things that need to be considered again as you're designing your study. So as I've been hinting at here, there may be some value that can be derived from additional targeted deep sequencing when you start looking at low-pass whole genome sequencing. And it's fairly self-evident, but you're still working pretty hard to find these rare alleles. They require a lot of sequencing in a lot of individuals. And that's a work in progress, and obviously, people are finding more every day, but in terms of imputation, they're still pretty hard to find. This particular figure is taken from an imputation service provider, GenCove, in one of their recent publications listed below there. And then we've layered some of our own data on top of that. And it looks, as you've seen in the previous talk, very similar. Again, just emphasizing the more coverage that you get, the better you're able to impute alleles. So that is the different colors moving from the bottom of the chart up to the top. And then from left to right, you're looking at the allele frequency in the population. And this is really, the crux of what I was talking about before. So if you're looking at 5% or above, then you're capturing, upwards of, 90, 95% of your non-reference calls in your imputed data sets, which is great. But once you go below that, particularly below 2%, the accuracy just drops off still, because we just don't have enough of those rare alleles in these current reference data sets. So in order to get down to 1%, for example, you'll need to look at tens of thousands of samples, honestly, in order to detect those 1% variants. And the other side of the coin is you start to run into problems with false positive rates as well, if you're relying on low pass data alone, when you get into those lower and lower frequencies. And this will obviously further impact your imputation accuracy. As you increase your coverage, you are able to pick up more variants in your low pass sequencing, and therefore also impute more variants at the end of the day with your low pass data. So we see that increasing, but it does start to provide diminishing returns again as you get to higher and higher frequencies and higher coverage. Where it really makes the most difference is in the low end of the spectrum, but even then, when you start looking at these low frequency variants in the 3 to 1% range, it's still really tough. So targeted enrichment then is a good way to go to help ensure that you're getting confident rare variant calling, and that you're capturing as much as you possibly can from a limited amount of deeper sequence data. So this figure just shows the range of options that you have available. That also represents the amount of data that's generated from each, and then what it can be applied to at the end of the day. So as we talked about with microarrays, they're obviously very affordable. They may be more limited to exploring common variants, but you can see a variety of applications below: GWAS, agricultural applications, polygenic risk score generation, as I mentioned, cytogenetics, PGX. All of these are still absolutely appropriate for microarray data. Also appropriate because, as we saw in the previous slide, low pass can give you very similar results. It can also start to open up more areas of the genome, again, depending on the frequency of those variants that you're looking at. So similar applications, but as you start to get more and more data, then you can start to also look at population health and screening of that health, and then go from there for health systems and screening for healthy individuals. But it doesn't stop there. So if you, for example, add an exome then to your low pass data, now you're actually looking at clinical grade detection. Exome 100x coverage is already the standard in the field. So you layer that on now and you provide both low pass with the targeted exome. Now you're really getting confident detection of those harder to find variants. You can start to look at biomarkers more seriously, and importantly, those that turn up in the exome often will have functional relevance as well, which may or may not be the case with what you're able to impute or pick up with your low pass data alone. So that's really the sort of minimum required for clinical diagnostics with that exome portion of the data. And then if we move in, obviously, to your standard 30x, 40x whole genome sequencing, that's still currently our best option for a high quality genome. You're going to get better clinical and all of your other benefits there. And also, obviously, for those other organisms maybe that don't have as much reference data available, and you're not going to be able to learn as much from lower data densities. So really exploring those non-human genomes is best done with the high quality, high depth coverage of whole genome sequencing. So that really, I think, covers most of it. But again, keep in mind, as the price of sequencing continues to come down and we get more of these 30x and 40x genomes, we learn more and more, we start to move across that spectrum that I showed in the earlier slides of the number of genomes available in your reference population, then that whole genome sequencing becomes less critical. You can actually start to move back to the left in this figure then and focus more on lower pass and potentially still enrichment with the exome, trying to pick up those harder to find variants. And with that, I think we're ready for questions and discussions. Thank you very much.
Thank you both for sharing your perspective on this topic. That was very informative. We had some questions come in, so we're going to move to the Q&A segment now. First question, this one's for both of you. What is the recommended read depth for low-pass whole genome sequencing?
Yeah, go ahead, Lisa, that's fine.
I was going to say, so, roughly anywhere between 0.4x to 4x is probably the norm. But we have seen, some people request like low pass sequencing, but actually go as high as 10x. I think it really just depends on someone's project goals and exactly what they're looking for.
Yeah, that's absolutely right. It all comes down to your specific needs. You can get away with it at quite low coverage depth, but you won't get as much information back, obviously, and that was sort of the last graph that I showed there. So that's one consideration. Also, how many samples do you have? Is it tens of thousands? Is it thousands? How much can you spend per sample? And how much data do you need from each? Because obviously you're going to have to sequence more to get more information, and that's going to cost more, obviously. So those are, I think, the main considerations as you're designing a study.
Dr. Yeager, you mentioned typical coverage for low-pass whole genome sequencing ranges from 0.5 to 10x. With regard to coverage, when do you see diminishing return, and what level of coverage is enough for imputation?
Yeah, so I think that goes back to the previous question a little bit, but it's really, again, your comfort level with the number of variants that are being detected. Is it enough to answer your question? Are there going to be some that are missed? So if you require really high confidence, then you're going to need more depth at the end of the day in order to achieve that. But again, keep in mind that you will not, despite that very high coverage, be able to address those rare variants in the population. So when you get below 5%, they're going to drop off. You're just not going to find them as reliably as those variants that are more common.
Dr. Louie, in your presentation today, you focused on human genomes. What other organisms are candidates for low-pass whole genome sequencing?
Yeah, so that's a really great question. So, if you think about imputation and everything that goes into it, it does require referencing a large set of sequencing data that's already existing. So candidates are really limited to some of these established workflows. So at GENEWIZ, we would limit the full workflow to either humans, dogs, cats, cattle, mice, corn, soy, and rice. Those are pretty much the well-established sets of data. Other organisms could be considered, but depending on the availability of any existing data sets, analysis options might be not quite like the full imputation that you would be expecting from some of the better established organisms.
This one's also for Dr. Louie. I'm interested in genotyping a set of samples, but the genotype is based on a specific gene target. Do you think low-pass would be a good alternative, or should I continue using PCR?
Yeah, so I think for that one, it just depends on your ultimate requirements. Depending on the level of coverage you're requesting for low pass, you may or may not actually capture that gene of interest specifically. That might be the gene that you're actually missing some coverage on. So if you need that exact SNP or that exact region covered to be able to make a call on your genotype, then maybe low pass won't be the greatest option. You might be seeking a more targeted option that you're currently using, especially if maybe the genotype that you're looking for has a high homology with another. It might be worth sticking to something where you'll get a physical confirmation. And if it's one of the genotypes or targets where you can have like an array to target it, it might be better. You might not need to do an actual PCR here.
Dr. Yeager, your slides discuss capture of a small percent of the rare variants. Is there a recommended coverage to capture most?
Yeah, for typical epsome coverage, 100x is the sort of minimum gold standard for that. Obviously, you can go higher. That will give you more confidence, again, with diminishing returns. But yes, to really have confidence, particularly if you're looking at clinical applications where you're going to be applying this information to a patient in some way, then you absolutely want to have a minimum of 100x coverage for your exome regions.
Dr. Louie, are there any differences in how samples should be prepared for low pass compared to the other methods?
So the quality and amounts for low-pass sequencing is pretty similar to that of other NGS options, like the regular whole genome or the whole exome. And I would say that low-pass does have more options to work with limited amounts of DNA. But in terms of like how to prepare the samples and the kits that you should be using, it's just your standard extraction types of workflows.
Both speakers suggested that low pass should not be used in the clinical space and that 30x whole genome sequencing is more suited. Can you elaborate more on why?
Yeah, I think that's actually a good point to clarify, because when you say clinical space, that encompasses quite a bit. So when we're talking about diagnosing rare variants in an individual that may have some functional effect on a particular protein or pathway, then that's going to absolutely be something where low pass alone is not relevant or appropriate. But looking at polygenic risk scores, for example, that's something that can be done with array data. You have many common variants that you can detect and use to compute that score. Adding in rare variants may make it more robust. Again, it depends on a variety of factors, your population, the particular phenotype that you're trying to capture or define, and then the prevalence of the various alleles that may be associated with that. So I think there's two ways of looking at it, Liz. You can elaborate further there.
Yeah, I totally agree with what you said. I also, similarly, the clinical space, that term is very general. So yes, if you're doing it for diagnostic purposes using low-pass sequencing at like the low coverages that we've been talking about might not be great because you could potentially get all these false positives and things like that. And I just think at a diagnostic level, the expectation is you want a physical confirmation of a specific sequence, especially if you're looking at a specific SNP or a specific variant. But there are labs that are using low pass more as a patient outcome testing, using as a risk assessment factor. But again, that is going to be preceding some deeper sequencing that would actually be used for more of that clinical diagnostic stage.
That's all we have time for today. If we have not gotten to your question, we will email you a response soon. Once again, I'd like to thank Dr. Louie and Dr. Yeager for their presentations and all attendees for joining us. If you'd like to view this webinar again or any other GENEWIZ webinar, the recordings will be available at genewiz.com.